Hi, everyone. It is a true pleasure to be here with you all today. And well, it would have been wonderful to have had the chance to meet in person. I am so pleased that through this virtual format, we are able to welcome participants from around the world. Really, the nexus of capacity and momentum when it comes to using genetics and genomics for malaria control and elimination is shifting. Every year, we are seeing more and more superb work coming out of malaria endemic countries, places like sub-Saharan Africa, the greater Mekong subregion. So the fact that we're having a free and virtual meeting that is accessible to scientists working all around those world and in those malaria endemic places is so important. Before I begin with my remarks, since this is technically my first GEM meeting, I wanted to briefly introduce myself so you know the perspective that I'm bringing to these remarks today. I have been with the Bill and Melinda Gates Foundation for 2.5 years now, and there I lead our surveillance, data, and epidemiology portfolio, working alongside our two senior program officers, John Cox and Esty Torak. The goal of our little corner of the Foundation's overall malaria team is pretty simple. We want to empower national malaria control programs to be able to make better decisions based on high quality, timely data. And whether that is crafting a new national strategic plan, whether it's developing a global fund application, or whether it's understanding whether the current crop of interventions that they're deploying is working, we really want NMCPs to be using data to inform how their program is run. That means we invest in things like improving routine surveillance systems and deploying new digital tools. It means that we support a number of geospatial and mathematical modelers to test out the potential impact of various combinations of interventions. And it means that we invest in new data streams that can provide actionable intelligence on a faster and a more accessible basis than things like an every few years parasite prevalence survey or an every 2 years treatment efficacy study. And one of those data streams that we're really helping to move from research into proof of concept move from research and proof of concept into a more routine operational usage within an NMCP. That data stream is parasite and vector genomic data. And my own background before joining the foundation was actually in pathogen genomic epidemiology. In fact, I was just thinking now, this exact time last month, I was here in this same virtual meeting hall running the bacterial and viral equivalent of GEM, the Applied Bioinformatics and Public Health Microbiology Conference. So pathogen genomics is where I come from. I did spend a decade working at the interface of academia and public health, really helping to roll out some of the first use cases for public health genomics. So while I may be a new face to some of you in this malaria genomic epidemiology community, I do have a long track record working in pathogen genomics as a whole. And what I want to share with you today are some reflections on what we've learned both in the past decade of scaling up pathogen genomics in settings around the world for routine usage, and a little bit about what we've learned from using genomics in the context of the COVID pandemic. I think both of these experiences hold a lot of lessons that are very applicable to what we do as we scale up malaria genomic epidemiology in endemic country settings. And there's 3 particular lessons that I wanted to specifically call out today ahead of the panel discussion. So the first point that I want to make is the need for integration. We simply cannot scale genomic epidemiology for malaria alone. If we really want genomic surveillance to be embedded within a national public health program, within an NMCP, we really need to work across disease verticals if we're going to ensure a sustainable future. From past experience, we've seen time and time again that when you put a sequencer in a lab and you say, you know, this is the TB sequencer or this is the foodborne pathogen sequencer, What often happens is that siloed sequencer ends up becoming a very expensive paperweight. It's not used enough by a single group. It's only funded with research and not operational money. You get personnel turnover within the group, and you end up with nobody that knows how to run the thing. And this is something that we really need to be proactive about preventing when it comes to the sequencing capacity that's being stood up for SARS-CoV-2 genomics right now. If we don't have a transition plan in place, if we don't have post-pandemic use cases for these machines, honestly, in a year or so from now, we're going to be able to really just open an expensive paperweight museum. So what you need is a cohort of trained lab technologists who can take extracted DNA from any source, run it on a sequencer that's being used multiple times every week or every month, and techs that can do all that basic upstream QC bioinformatics work, like making sure the run actually worked. You basically need a small pathogen genomics core facility that can handle malaria, TB, HIV, viral samples, you name it. The sampling platforms are going to vary from disease to disease, as will the downstream bioinformatics analysis and interpretation steps. But really, if you want to achieve the economies of scale that make sequencing feasible, and if you want to retain a skilled workforce, you really have to think beyond a single pathogen and have a machine that is serving a range of use cases. If we were actually on site at the Wellcome Genome campus, at this point, I'm probably wave my hands around and say that one of the reasons that the UK's COVID genomics effort, COG UK, has been so successful is because a core facility with a cross-pathogen mandate, like the Sanger Centre, or like the Public Health England genomics team at Collindale, those things existed. Those core facilities existed and could be quickly leveraged for SARS-CoV-2 work. And I can tell you that the converse has also been true. Many public health units in North America who had been doing some sequencing for a very specific pathogen, they struggled with the pivot to COVID genomics. And I should underscore, I'm not talking about academic research groups here, but rather, a city or a state or a provincial laboratory. These were public health units that treated genomics for the most part as something that fell in the domain of research rather than surveillance. So any genomics work that they were doing tended to be highly project specific. Lab techs would be reassigned to other work once the sequencing project had finished. All of that really made standing up a rapid genomics response much harder in these sorts of groups. Genomics had been done in a silo, so essentially these groups were starting from scratch when it came to their COVID work. And when you have to start fresh, when you have to train new people, order new reagents, work through data management and data sharing issues with every new project, it makes sustainability really, really challenging. So I think the malaria community, as we look towards a future where labs like the nodes in Africa CDC's Pathogen Genomics Institute are routinely working on malaria sequencing, that needs to be a future where that malaria work is being done alongside genomic epidemiology of a range of other pathogens as well. I think that really is the key to unlocking efficiencies in the system. The next takeaway that I want to leave you with is the need for us to work on the upstream and the downstream challenges when it comes to malaria genomic epidemiology. I often liken genomic epi to a bow tie. The sequencing itself is the knot in the middle because it's really tight and it's tidy. We've figured that part out for the most part. The ends of the bow tie, though, the floppy bits that are sometimes completely sideways, depending on how good you are at tying a tie, those are the upstream challenges, like figuring out sampling frameworks and solving issues with equipment and consumables procurement. And then those are the downstream challenges, like common approaches to analytics and issues around data and information sharing. And it's time that we really get the ends of the bow tie sorted out and to get our ties tied properly. I think, these are both very big issues, very big domains, the upstream and the downstream. But I think upstream, the most pressing issue that we need to be looking at is ensuring that scientists in LMICs have equitable access to equipment and reagents. That means paying the same price, or better yet, a subsidized price as compared to other buyers. It means not having supplies held up at a customs warehouse where they might spoil or be subjected to excess duties and tariffs. It means being able to have a service technician come visit their lab site and make any necessary repairs on a timely basis. And this is an area that the global health community is certainly working hard on, and which COVID has really accelerated. But we do have to protect the gains we've made, and we do have to go a lot farther. Part of this, I think, involves working directly with sequencing companies and with reagent suppliers and really being dogged about it. I can tell you that for many years, a lot of companies just simply didn't see markets like Africa as priorities. And we are starting to see that change. But we as a community really can't let up pressure. And I'm sure that many of you who work in academic units in places like North America or the UK or Europe might be wondering what you can do versus what an entity like Africa CDC or the Gates Foundation can do. But the answer is really simple. Make sure that the company reps you work with know that this is a problem. Advocate for better market access for your LMIC friends and collaborators, and invite LMIC scientists to speak at the conferences, the meetings that these companies are attending, so that they can see for themselves the really, really good work that's happening there. On the larger global architecture stage, this is certainly an area of tremendous interest for us at the Foundation, for the Global Fund, for USAID and its constituent entities, and for groups like Africa CDC and the African Union. There are a lot of moving parts here in domains from advocacy to market access, but it is something that people in this field are committed to improving, and it's something that really does go beyond malaria to include a range of other disease communities. On the other end of the bow tie, that was the most pressing upstream challenge. I think one of the most pressing downstream challenges is around bioinformatics and data sharing. And this is one that I really think is going to unfold differently in different pathogen areas. So here, malaria really is going to have to chart its own course. I think that the past and the recent history of pathogen genomics show us just how much variation there can be within this space, depending on the nature of the disease in question. So for an example of where open data sharing, open analysis is very much the norm, I think the FDA, the Food and Drug Administration's Genome Tracker project provides a really, really nice instructive example. Genome Tracker collates bacterial genomic data and very detailed sample metadata for a number of food and waterborne pathogens. And what you see are public health agencies from around the world, the US, Canada, the UK, Europe, they are contributing their data in real time, and they typically both make raw FASTQ files and assembled consensus sequences available. And if you stop to think, why is there so much buy-in to this particular project? I personally think that it boils down to a few things. First is that foodborne outbreaks frequently cross jurisdictional boundaries. You can have a food item being recalled in nearly every American state and Canadian province. There's plenty of examples of outbreaks spanning the UK and Europe, from Salmonella to E. coli. It's not like a respiratory pathogen like flu or like SARS-CoV-2, where you can lock your country down, close the borders, and stay safe. These outbreaks always span multiple regions, so that international data sharing is vitally important for tracking and solving these outbreaks. And I would argue that, like foodborne illness, the same importance of international data sharing is true for malaria. Whether you're a country who shares borders with other endemic countries and sees a lot of population mobility, or whether you are a near elimination setting, trying to understand whether your handful of cases are local or imported, being able to see the data from other settings provides really, really critical context. I think the second reason that something like Genome Trackers had a lot of buy-in is that there's a long history of formal data sharing networks in that space. Before whole genome sequencing came along, the foodborne microbiology community was using pulse field gel electrophoresis. They were routinely sharing patterns through the PulseNet network. So routinely sharing whole genome sequencing data wasn't really a big stretch for them. And again, I think this is an area where malaria is well positioned for success. MalariaGen, for instance, has set a superb precedent for sharing both Plasmodium and Anopheles genetic and genomic data of various types, and has done a lot of work establishing data ownership and data sharing agreements that I think can be adapted by other groups outside the network. I think one of the third reasons, and perhaps this is the most critical, why genome tracker and foodborne data sharing has been a success, but we haven't seen that play out in something like influenza or SARS-CoV-2, it's that when it comes to food and waterborne diseases, these are areas that are just not fraught with IP issues. They're a domain where, because there are no real treatments or vaccines, there isn't inequity in terms of the people that are generating the data not being the ones who then benefit from the therapeutics that are derived from that data. This is a huge injustice in the COVID-19 space, and it's been an issue with respiratory viruses going back to highly pathogenic avian influenza in the 1990s, but it's not something that's really affected the foodborne space. We haven't seen it play out in other diseases like TB, which I worked in for many years, and where again, there's fairly friction-free sharing of genomic data. So in malaria, I have to say, I honestly don't know how this is going to pan out. I think the dawn of the mRNA vaccine era is going to draw a lot of attention to the potential for deriving IP from shared genomic data. And I think it would really be great for us as a malaria genomics community to be proactive in setting out the principles that should guide data sharing in this space. And I think there's a huge role for organizations like what used to be Plasmodium Diversity Network Africa and is now PGDNA. I think there's a huge role for groups like that to play in setting those norms. I'm going to end with the third takeaway, and I will be brief here because it's very simple. The most successful public health genomics efforts involve teams that span different domains. In any given setting, whether it's elimination or high burden, we need to ensure engagement across the board, from academia to government, and we need to work across areas from surveillance to functional genomics if we're going to have a really cohesive and effective malaria genomic epidemiology program, sorry, not a problem. We don't want a problem. I'll give you two examples from COVID genomics where sort of taking this cross-sector, cross-domain approach has worked really well. The first, I might get some eye rolls for this one, but hear me out. The first is the recent genomic surveillance work in the US. And certainly the US does lag behind other places when it comes to COVID genomics, especially early in the pandemic. But more recently, we've ended up in a really interesting place where sequences were coming from the usual state public health labs, but they were also coming from academic groups, from hospital systems, and even from industry partners. And the system certainly wasn't without its challenges, especially around information sharing. But I do think there is something perhaps to this hybrid intersectoral networked approach to genomic surveillance that might be worth exploring for other pathogens. And the second example I wanted to quote was the superb work that's being done in South Africa to not just track emerging SARS-CoV-2 variants, but to really understand the implications of those variants. The genomic surveillance team there works very closely with functional genomics and with immunology groups who can really explore how these variants differ from wild-type viruses, and they're also very closely tied to groups leading vaccine trials, so they can look at the impact of variants on vaccine efficacy. And it's, I think, a really nice example of an interdisciplinary team being able to offer a really holistic and complete view of what is an ever-changing infectious disease scenario. I shall end there so we can keep plenty of time for the panel discussion. And there, I would love to hear your questions and your thoughts on what we could do best when it comes to operationalizing malaria genomic epidemiology. I know that part of this session title is Barriers to Translation, so I would love to hear what you think the biggest barriers are and what you have in terms of bright ideas for solving those. Thank you so much. It's been a pleasure giving these remarks, and I really do look forward to the panel discussion.